In life, all good things come to an end, and our immune responses are no different. We developed these cells, we built these cells, we put them to work, and now it is time to end them. The immune response, as we've seen in kind of the previous videos, is a deadly and inflammatory tool, and continued inflammation is a bad thing. So when I've talked about it in general, I'm talking about it kind of in the context of a pathogen, right? You get sick and you mount an immune response, and that immune response grows and goes up and up and up. And then eventually, if we don't stop it, it's going to continue to cause disease. And at this point, that's when we move into the realm of chronic inflammation. And chronic inflammation is very bad. Whether or not that chronic inflammation is directed toward a pathogen that we can't get rid of, like a chronic viral infection, or a self-antigen, as would be the case in like autoimmune or hypersensitivity disorders, or even an innocuous antigen like an allergy response, inflammation leads to pathology. Your pathology discipline director and I often kind of talk about this, that so I teach you immunology, and immunology is to math as math is to physics, where pathology equals physics. Okay, so if immunology is math and pathology is physics, immunology is just applied, or pathology is just applied immunology. That's a really complex way of saying my cells create his pathology. And that's pretty much the whole concept here. So what we want to do is avoid pathology, right? So there becomes a point where the immune response is not normal anymore. The pathogen is gone and it's time to shut things down. And as the immune response returns to normal, that is referred to as the homeostatic state. And that's kind of what your immune response is trying to maintain all of the time. And there's normal turnover in your immune cells, certainly. And at the end of an immune response, your body wants to go back to having kind of the normal number of cells in it. This largely occurs because as pathogens die, the lymphocytes that respond to them no longer receive the survival signals that they need. So if a lymphocyte is not triggered, if it's not getting kind of constant positive reinforcement, eventually it kind of dies off by neglect. And that's one way. But there are also some more kind of direct ways the immune response kills itself off. And I think that's kind of one of my favorite things about this particular part of the immune response. It's kind of like a sudsy soap opera. We're going to move from like fighting to betrayal, because at this point it's I made you and I will take you out. And that is kind of how the immune response is looking at many of the soldiers in its army at this point. There is one golden parachute for immune cells that want to keep living, and that's called memory. In every immune response, there are some cells that are triggered to kind of be more long-lived, that they're going to go the extra distance and they're going to live for decades. And we're going to talk about that in a later video. Okay, so these are kind of the four components of an immune response, and we've already talked about these. So recognition, when antigen meets antigen receptor, or T cell meets antigen, or B cell or antibody, whatever. This is the recognition of the pathogen. Recognition leads to signaling, which causes clonal expansion, which is your amplification stage, and as well as differentiation. And that's particularly important, right, for our CD4 helper cell populations, right? We've got those Th1s, Th2s, Th17s, whatever, as well as our B cell population, right? Because we want to be able to make IgG, IgA, IgM, all of that. And once they've differentiated into it, they're going to be able to carry out their effector responses, right? So that would be actually making IgG, IgA, IgE, or being a Th1 cell versus a Th2 cell, and the cytokines that that cell then produces. We're now going to move on to this section, regulation and control. So After a pathogen has been defeated, we have to stop the immune response, and it's controlled by a bunch of different regulatory processes. And these regulatory processes are kind of grouped under the concept of peripheral tolerance. So the last time we talked about tolerance, we talked about central tolerance. And central tolerance is what happens in the primary lymphoid organs where T cells and B cells are educated on how not to respond to self-antigens, right? And if they can't be convinced not to respond to self-antigens, they're killed. Peripheral tolerance is kind of a wide range of mechanisms that help to promote contraction of an immune response, but also kind of in the day-to-day life when there is no, mass expansion of immune cells that was created in response to an antigen to contract, just kind of try to keep that homeostatic level that we want in our immune response. All right, so let's talk about immunologic tolerance a little bit. So at this point, we have this fully developed immune response that is doing major damage against the pathogen. And as a result of that, we're getting bystander damage against our own tissues. And that's through, neutrophil activation, macrophages, complement, all sorts of things. So at this point, we need to stop a train that is speeding down the track with a broken brake. And the only way to do that really is by implementing tolerance mechanisms that kind of keep everybody under control in the first place. That's going to slow down the immune response and eventually lead to death of the majority of the cells that responded. Because remember, death is kind of the immune response's default response. So there are three main mechanisms that I'm going to talk about as part of this. Clonal anergy or exhaustion. Those are one, okay? T regulatory cells or Tregs. You'll hear me refer to them as that. And then clonal deletion, which is basically like we're going to kill all the cells, all right? These three mechanisms all work together to ensure the end of an immune response and will largely result in the death of the cells. When these three mechanisms aren't working perfectly, that's when we wind up with the result of hypersensitivity or autoimmune disease. So that's why I say that basically the response for ending an immune response is the same as preventing autoimmunity, because what you need to do is think about how are we seeing the antigen, and that's kind of what this figure is showing. So remember when I said that all immunogens, this was a while ago, all immunogens are antigens, but not all antigens are immunogens. And that's kind of the whole concept behind tolerance. So there are a couple things that determines whether or not an antigen is actually an immunogenic antigen. So if it's a self-antigen, that basically means that the location of the antigen, whether or not there's co-stimulatory molecules present when the effector cell is seeing the antigen, and how long-lived or persistent that antigen exposure is, might tell the body, you know what, this one's supposed to be here and we should stop attacking it. Whereas an antigen that shows up when there is an inflammatory response will actually induce co-stimulation, which will actually make it more immunogenic. And that's kind of what I'm trying to get at here, is that when we're thinking about peripheral tolerance and we're thinking about mounting an immune response, so that would actually be over here, so we want the immune response, versus stopping one that already occurred, avoiding hypersensitivity, or just avoiding inappropriate responses in our normal everyday life, we kind of need these things to be in balance of each other. We need it to be that if the antigen is immunogenic, there's a big enough splash made by our inflammatory cytokines like TNF-alpha, interferon, IL-1, IL-6, and that, co-stimulation is present, that the body actually stands up and takes notice. but not so much that it would overpower a don't attack signal that might come from any of these mediators. So we're going to talk about those mediators a little bit more in depth. Okay, so in case I didn't define it, peripheral tolerance is literally any mechanism occurring outside of the central lymphoid organs that limit the activity of an immune response. All right, so we're going to start with differentiating two really important forms of peripheral tolerance: clonal anergy and clonal exhaustion. They're not the same thing, even though they kind of have the same end result. All right, and in this, I'm also going to throw in describing no co-stimulation, because that's the other kind of way we get here. All right, so first off, let's define anergy. Anergy actually, I think, is relatively easy to define. You know what energy is. We have energy to get up and do things, okay? That's probably, physicists in my viewing audience are probably rolling their eyes right now. But energy is, you have the ability, the function, the calories, whatever, to get up and actually do something, perform a process. Energy is the opposite of that. When I think of energy, I think of my son. 1 minute he has tons of energy and is running all over the house and kind of driving me nuts, even though I love him. The next minute, when sometimes maybe I need a break or need to get ready for work, I plop him down in front of the TV and put on his favorite movie, and then I could literally throw things at his head and nothing would happen. He is anergic. He is unable to be stimulated. My predecessor used to call anergic cells zombie cells. It doesn't matter what is done to these cells at this point, they are not responding. All right? So let's break this down even further. When we're initially priming a T cell, you have your signal 1, right, which is TCR and antigen. Signal 2 is co-stimulation, right? Co-stimulation happens when B7 molecules on the antigen presenting cell meet with CD28 on the T cell. All right? Now, what if B7 wasn't there? So all we had was signal 1, antigen and antigen receptor, and CD28, but no ligation of that CD28. That is no co-stimulation. And when that happens, basically the T cell gets the instruction, not you. You don't respond. You could be dangerous, because if you were something that was supposed to respond, there would be an inflammatory signal that would have upregulated B7 on this antigen presenting cell. And it's not there. So you're never going to respond again. And it just kind of shuts it down. And it doesn't die right away, but it can still move around the body. It just can't respond. It's a zombie at this point. So that's no co-stimulation. There's another way to get to anergy, and that is the work of the molecule CTLA4, okay? So anergy can happen a couple of ways. So I showed it over here with this is no co-stimulation, all right? This is what we expect to see, CD28 binding to B7, signal 1, signal 2, proliferation. Good. Now, as the immune response goes on, another molecule comes up to take the place of CTLA4, to take the of CD28, to steal the attention away from B7, and that's CTLA4. As cells become more and more activated, the T cell will now start co-expressing CD28 and CTLA4. This is kind of an internal control for the T cell. CTLA4 is able to bind B7 molecules with 20 times higher affinity than CD28. So that means that as the immune response kind of settles down and there's less inflammation and therefore less B7, the B7 molecules are going to preferentially bind CTLA4 more than they're going to bind CD28. And CTLA4 is an inhibitory molecule. So it basically tells the T cell, become anergic, shut down. This happens in acute events and at the end of an acute immune response. This is something that happens within days to weeks of an infection. That's different from the concept of exhaustion, which is what I'm showing down here. The marker for exhaustion is PD1, okay? Exhaustion is how, if you've ever had a project that just, you had to work on it so much that, it got to the point where you almost kind of didn't care how it turned out because you'd been working so hard and you just wanted it to be over. That's exhaustion. Exhaustion happens to cells when they have been exposed to their antigen over and over and over and over again for months to years. This is the kind of marker that shows up in tumors, in chronic viral infections like HIV. Those are kind of the places where we expect to see it. So what happens is this marker, PD1, which is also known as programmed death 1 molecule, is found on the surface of cells that have been being activated at a high level for long, long periods of times, months to years. That's kind of key, okay? And what happens is it binds its ligand on the antigen presenting cell, PDL1. And that basically tells the cell, you're exhausted, you need to sleep. Just stop. Stop responding. Stop, stop, stop. All right. And it kind of stops it from being able to make any sort of response. Why I tell you about CTLA4 and PD1 is that these guys have actually become targets for immunotherapies, particularly in cancer. There's anti-PD1 and anti-PDL1 monoclonal antibodies now that are used basically to block this pathway. And when that pathway, when this receptor ligand interaction gets blocked, that T cell continues functioning and it begins producing cytokines and performing cytotoxic functions again, which obviously in cancer we want. Okay, so I've summarized this in a table, and this is a table I'd spend a little bit of time on because I kind of expect you to be able to do this. So exhaustion occurs when there's been excessive continuous stimulation. The signal received is a persistent overstimulation, and then PD1 is upregulated on the T cell and bound by PDL1 on the antigen presenting cell. In this, we're talking weeks, months, years. These are prolonged antigen exposures. The cells aren't really going to proliferate and they're going to have very little to no effector function. CTLA4 can happen in one of two ways. There's either suboptimal stimulation or there was stimulation and now it's kind of waning, so CTLA4 is coming up. So you can get energy when you have signal 1 in the absence of signal 2, or because CTLA4 has come up and it's the shiny, it's the prettier bird, it's the shinier package that B7 wants to bind instead of CD28. This one is going to come up within a couple of days of the start of the immune response. Again, we're going to have very low proliferation from these cells and they will have no effector function because they're zombies. All right, we're now going to talk about my favorite cell in the entire immune response, and those are the T regulatory cells. I literally wrote my thesis on T regulatory cells. Don't worry, I'm not going to make you read it. But we are going to talk about the amazing functions of T regulatory cells. T regulatory cells are the sit down and shut up T cell of the immune response. They tell everybody when it's enough and you just need to stop because you are now messing this whole thing up for all of us. That's kind of their whole deal. And they have a lot of ways of doing this. So most of it, because these are CD4 positive T cells, most of it is through secretion of mediators, okay? So the first thing is inhibitory cytokines. I've talked about kind of cytokine milieu and the microenvironment of cytokines a lot. And if you have kind of an inflammatory cytokine response, that's going to overpower anything the T regulatory cells could do, and you're going to get more and more inflammatory cells. But what happens is As some of those cells become anergic and exhausted or deleted, as we're going to talk about in a minute, the microenvironment of the immune response becomes more and more tolerogenic. What I mean by that is that there are higher concentrations of inhibitory or tolerogenic cytokines. The main ones are TGF beta and IL-10. The jury kind of goes in and out on whether or not IL-35 actually is a cytokine or is real, and if it is tolerogenic. I think currently people are very certain that it is real. We'll see if it sticks this time. It's come back and forth a couple of times in the last decade. And I think there's also a new one now, IL-37. So we'll see if that one joins the party. But either way, what you need to know is that T regulatory cells are pumping out IL-10 and TGF beta, which is kind of like, I don't know, prescribing like a calming drug to the entire room of cells. It's just going to calm things down, stop things from proliferating, stop cells from differentiating into inflammatory effector cells, just kind of tell everybody to keep it copacetic. It's, turning on the lights at the end of a party and switching to serving water and soda. Okay. The other thing they can do is they can target antigen presenting cells. So remember, antigen presenting cells actually prime T cells. So in this case, we're actually going to go to the antigen presenting cells and say, hey, you, stop making those T cells so good. They're doing too good a job and they're damaging everything. So the way they do that is a couple of ways. First off, they have IDO, indoleamine 2,3 deoxygenase. Now, I'm not going to go through the whole pathway of this. Your biochemistry professors can do such a better job of this. But really, they produce a lot of IDO, which basically messes up tryptophan metabolism. And when cells don't have good tryptophan metabolism, they aren't able to proliferate and make proteins and carry out effector functions. So this is it's almost like a cytokine. It almost kind of belongs over here. It's another thing that's just going to kind of put a damper on the whole immune response. T regulatory cells also have high levels of CTLA4, so they bind up some of the B7 on the antigen presenting cells, so it's not available for effector cells. They have this other molecule, LAG3, which is actually a CD4 homolog, so that actually binds up the MHC class 2 on the antigen presenting cells so that it's no longer able to bind TCRs on effector cells. And it can actually do this at very low ratios, like 1 T regulatory cell is actually pretty potent for this. But T regulatory cells don't just rely on antigen presenting cells to starve out cells. They're really great at starving people all on their own. So one of the main markers for T regulatory cells is CD25. CD25 is the IL-2 receptor alpha chain. And if you remember my video on IL-2 receptors, you know that IL-2 is lymphocyte food. And the IL-2 receptor alpha chain specifically is a tryptophan nap-inducing top button of your pants unbuttoning Thanksgiving feast. This is the thing that T cells need to really chow down on that IL-2 and proliferate like mad. But CD25 is really highly expressed on T regulatory cells. So they basically gorge themselves on all of the IL-2, and then there's none left for the rest of the cells. They do the same thing with adenosine. They actually have high expression of CD39 and CD73, which are the adenosine receptors. So they soak up all the adenosine, and once again, the cells can't grow or anything. There is also some indication that they have some cytotoxic function, that they can produce granzymes and attack effector cells. That data in humans is a little bit sketchy. So really it's more about the mediators, the non-inflammatory or tolerogenic mediators. All right, this is the last mechanism. This is where we're gonna kill. We're gonna kill almost all of the cells, all the effector cells, because we made way too many, and we needed to because we had to kill that pathogen. But now the body can't even possibly sustain all of these effector cells, and these effector cells are really rowdy and making a ton of inflammation everywhere. So we have to stop them. So there's two mechanisms of clonal deletion, and clonal deletion really just means we're going to kill all of the cells of a specific clone. So remember, a clone is basically, you've got this TCR that say is specific for light blue antigen. It got, signaled, activated, primed, and it made a whole bunch of itself, right? Because there was a whole bunch of light blue antigen that we had to kill. But now we have too many, so we're going to kill off almost all of the clones of this T cell and maybe just leave like a couple, okay? All right, so the first mechanism of clonal deletion is one we kind of already talked about. It's antigen recognition without co-stimulation, but it's like really, really, really strong antigen recognition. This is if it recognizes its antigen so strongly, it kind of gets overpowered, and it's like a short circuit that then just kind of fries the cell and induces apoptosis. In this way, we're going to keep newly created naive T cells from becoming activated as their response is no longer necessary. So, and this is also kind of a fate of even anergic cells that eventually, even though they've first gone anergic, at some point without any sort of stimulation, they're going to die off. This is actually the more common way to think about clonal deletion, and it's known as Fas ligand, okay, or CD95. This is probably the most important mechanism of peripheral clonal deletion. In this mechanism, a cell decides to self-sacrifice. The cell has decided to die, and it can't really kill itself, but it will instead rely on killers of the immune system to kill it for them. So basically, the target cell, this cell decides, you know what, I've had enough, it's time to go. And it will express the Fas receptor on its surface, okay? And then a nearby CD8 positive cytotoxic T cell that expresses the Fas ligand will come and basically put the cell out of its misery. When Fas ligand binds to Fas receptor, there's a death domain in the Fas molecule which is engaged and leads to activation of the caspase cascade. And that basically leads to apoptosis. There is actually one other way this could happen. The one exception is that CD8 positive T cells can actually kill themselves this way. So the same CD8 positive T cell could already have Fas ligand on its surface and then could upregulate the Fas receptor and go, and then that's it. There are children who have mutations in Fas ligand or caspases, which are necessary to accomplish apoptosis, and they actually develop an autoimmune disease associated with lymphocyte accumulation. These human diseases are collectively known as autoimmune lymphoproliferative syndrome. They're pretty rare, thankfully, and they're the only known deficits in apoptosis that causes autoimmunity. And those are the peripheral tolerance mechanisms.